So I'm going to bring up this screen because it's the one that we left off on the last time that we were writing things down on Thursday of last week. Chances are you have already written this unless you happen to have not been here on Thursday. But in any case, I just want to put you in the right frame of mind for talking about enzymes. The lab activity that we did on Friday was to help illustrate to you what could affect how an enzyme functions. And so to know what could affect how an enzyme functions, we need to know what an enzyme normally does, what is supposed to happen. So I highlighted a few words here that I'll just highlight again. For example, active site and substrate are both words that apply when we're talking about enzymes. And we'll discuss those a little bit further, but I wanted to highlight them here. Now, in the video that I made the last time we talked about this, I showed you a little animation that illustrated what an enzyme really did. The idea is that an enzyme helps two molecules, in this case sucrose and water, meet up so that water can help perform hydrolysis, which is the reaction where water breaks a larger macromolecule into smaller pieces. A couple of things that I want to point out because they're really important is that you can use an enzyme over and over and over again. It's not actually being used up by the reaction. As soon as it's done helping sucrase number 1 and water molecule number 1 react, it can move on to the next thing. Now, does anyone have any questions that they'd like to ask about enzymes at this point here? Because I'm going to give you a bit more details about what actually happens in the next part. All right, so stuff that's new, and I will make this bigger, is that when we talk about how an enzyme functions, we are talking really about something called the lock and key model. Now, this isn't a steadfast name that you have to use, but it's a good analogy for what's happening with an enzyme. In the lock and key model, the enzyme is the key and the substrate is the lock. And so what we're trying to say is that an enzyme can unlock the substrate, but it won't open the door. So if you think about whether it's easier to open a door that's locked or easier to open a door that's unlocked, I hope you would say it's easier to open a door that is unlocked. So an enzyme is something that will unlock the substrate. It's not actually making it do the reaction. So if I continue with the analogy, it's not opening the door for me, it's just unlocking it, making it easier to open. And so that's why this is a good analogy for what an enzyme is doing. Here's some points about enzymes that are true. If an enzyme is going to catalyze a reaction, it absolutely must come in contact with the reactant molecules. If the enzyme can't touch them physically, then there's no way it can act like a key. There's no way it can make this molecule have a reaction more easily. Catalyze, if you remember from the previous screen, means speed up a reaction that already occurs. Now, when the enzyme comes into contact with the reactant molecules, and the reactant molecules is what we would call the substrate, the compound is called the enzyme substrate complex because they're all together. They didn't turn into one molecule. They're still two separate things, but they're physically attached to each other. Now, where does the substrate attach? The substrate attaches to the enzyme at the active site. So an active site is the spot on the enzyme that's the same shape as the substrate molecule. So if you look at my silly illustration down here, you'll see that in my picture, I have nice goobery, fingery, gooey shaped enzyme and a substrate that's exactly the opposite shape. Honestly, any shape will do here. If you don't like drawing, much like I don't, that's why I borrowed a photo from somewhere else. The key thing to understand is that the active site is exactly the complement to the shape of the substrate. They fit together like pieces of a puzzle. Does anyone have any questions about what the active site is? So the substrate attaches to the enzyme at the active site of the enzyme. Either the substrate will be broken up, and since we're coming at this from the context of digestion, that's usually what we're talking about. So either the substrate is broken up, which is called catabolism, or the substrate is attached to another substrate, like I'm putting them together. That's anabolism. If I wanted to be general, I could just talk about metabolism. That's not saying whether things are being broken down or put together. That's just being very general. So a way to remember, and I remember this only because anabolic steroids is a terminology that I've heard in the media before, and chances are you have too. When people take anabolic steroids, that's usually when they're trying to build up muscle, and so that's the association I have in my head that helps me remember that anabolism is putting things together, and catabolism is taking things apart. Does anyone have any questions about the two kinds of metabolism we're mentioning there? Just so that we can make a link to words that we've already used. In our context, when we broke things up, we performed hydrolysis, because we've been talking about macromolecules, and this is the process they undergo. And when I attach a substrate to another substrate, in the context we've been discussing, this would be dehydration synthesis. Now the last thing about sort of the shape is that most enzymes have a very high substrate specificity. Not perfect, but high. And so on purpose, I didn't write that it was 100% or that it was totally all the time true. I'm just simply saying that most enzymes are quite specific for their substrate. In other words, they will attach to only one type of compound. What that means is that if two molecules have basically the same shape, they might both be able to attach to the enzyme, even though maybe only one of them is supposed to. This idea is how a lot of drugs work. A lot of drugs that are taken recreationally or medicinally, it's the same idea, work based on this principle. A lot of things are the same shape as a molecule that they're trying to mimic. And so some drugs are the same shape as a molecule that's already in your body, and they will perform the same effect. And so any sort of stimulant drug that you could take generally would be doing something like that, where it would be acting like a substance that's already in your body, but because now you just took a bunch of it, it's sending that signal a whole bunch of times. On the flip side, I could use a substance that would block an enzyme. It might have almost the same shape as some molecule I'm trying to break down, but there'll be something about it that's different. It will be different enough that the enzyme won't be able to break it down, and then the enzyme won't be able to break down other substances either. And so this idea of substrate specificity is high, but it's not perfect. So things could go wrong if they're the same type of compound as one that already exists in your body. And this is all dependent on the shape of the active site. the more specific the active site is to a particular molecule, the less chance that some other type of substance would be able to attach to the enzyme. Does anyone have any questions they'd like to ask about any of that information? So what I'd like to do next is show you what actually happens when those two things are attached to each other. We know that either they're going to be broken up or they're going to be put together, but we need to know a little bit more about why. So let's make this a little bit bigger. Oh, that's way too big. Let's just do page width. So this is the same picture. I'm not drawing something new. I'm just using the same picture to continue this. When an enzyme is performing its function, what's actually happening? The idea is that the presence of an enzyme will speed up the reaction. So that's a key point. Reactions happen faster when enzymes are around. The reason that this happens is because the active site makes chemical change easier. And what that actually means is that I lower the activation energy. Anytime a chemical reaction is going to happen, you need a little bit of energy to get it going. Even if it's a reaction that's going to produce energy in the long run, you always need to give a little bit of energy to get things started. That's the activation energy. So every reaction requires a certain amount of activation energy, energy to get it going. This is the energy required to break a bond or to make a bond. So if I want to start a chemical reaction, I got to start somewhere, and that always takes a little bit of energy. So if you think about what happens when you put the key in the ignition of your car. When you put the key in the ignition of your car, you have to have a little tiny bit of battery power to get the spark that's going to ignite the fuel. As soon as you get that going, your alternator recharges your battery and it's ready for the next use. Let's compare that to this. It's kind of the same idea. Without the battery, you couldn't start your car, even though you really don't need the battery to make your car run all the time, unless, of course, you have an electric car, in which case I don't really know how that's all working. But in an internal combustion engine automobile, that's the idea. Activation energy is like that little bit of battery power you need to get started so that your actual reaction can happen. So if I take a look at the illustration, this is illustrating catabolism, where I'm breaking something apart. My bonds are being weakened when the enzyme and the substrate are attached, and this just makes it easier for my one substrate to become two different products. So it's broken apart. So, can an enzyme make something happen that wasn't already going to happen? No. It just makes it easier for something that was already going to happen, just makes it easier for it to happen. Does anyone have any questions about the description of an enzyme the way it is up here? So aside from what an enzyme does, we need to know about the things that we can do to an enzyme to make things happen differently. Now you already know about proteins becoming denatured, because that was general to proteins, and enzymes are just one of the things that proteins can do. What we're going to talk about now are things that are specific to enzymes, things that will affect how an enzyme functions. Does anyone need this screen to still be up? Okay, we'll just wait for a second. So the thing we'll talk about the most are things that block enzymes. And then we'll go through a short list of other things that could affect the way an enzyme functions. Looks good over there. So we're going to talk about two kinds of inhibitors, competitive and non-competitive. Now I have some illustrative examples here. Don't feel like you need to copy every detail or memorize every detail of the illustrative example. They're just there so I have something real life to talk about. I'm not good at drawing, and if that's the case for you too, keep in mind that this is on Google Drive in a folder that I shared with you and sent you an email about. And your textbook has equally good diagrams that represent these things. The first kind of inhibitor is a competitive inhibitor. An inhibitor by nature is something that's not going to help an enzyme. So all inhibitors are going to reduce the function of an enzyme. What we're going to specify is how they're doing that. A competitive inhibitor is a molecule that is similar in shape to the substrate molecule, and it binds to the active site. That is the key thing. That is the thing that makes a competitive inhibitor different from a non-competitive inhibitor. This point here. It binds to the active site. If it's the same shape or a similar shape to the substrate molecule and it binds to the active site, well then it's preventing the substrate from attaching. Literally, there is competition for the active site's heart, and the competitive inhibitor is winning. So the first illustration is trying to show you how if my substrate molecule looks kind of like that, it's got a circle-y thing on top and a triangle on the bottom. It should be able to attach to an enzyme. So there's substrate, there's enzyme. It should be able to attach to an enzyme that has a triangle-shaped hole in it. When it does, activation energy is lowered, and now my substrate breaks apart into two pieces. So this would be the progression. It attaches, and it breaks apart. That looks exactly like the picture on the previous screen. When I have inhibition, another molecule, my inhibitor, attaches to the enzyme. It has the same active site binding shape, so it has that same triangle piece, but the top of it is a different shape. So it will bind to the active site, but it will not get broken apart. It'll just sit there and block my actual substrate. So right here, it's blocking my actual substrate from attaching itself to the enzyme. That is a competitive inhibitor. So I've got a real-life example of something that does that. Perhaps you might be aware of something called sulfonamide drugs. Think things like Pepto-Bismol. They have some sulfur in it. They're things that reduce nausea. In particular, they used to be prescribed to pregnant women who were having morning sickness. Now this is way back in the day, before my day too. And at first it seemed like it was like a miracle. Oh look, all these women who had morning sickness are no longer sick. But after the fact, and babies were born with defects, realized that that drug, although it made you feel better, was inhibiting folic acid synthesis, and that's something that you need for proper development. In particular, babies were born, say, without arms or without part of a limb due to this. And so that would be an example where the drug was a competitive inhibitor for the enzyme that was helping make folic acid. Another example of something that's a competitive inhibitor would be something like venom. Now the actual mechanism here I would not expect you to know. If you take Bio20, you'll learn about this molecule called acetylcholine, ACH. It is the molecule basically that lets your nerves and your muscles communicate and tells your muscles do something. Now a molecule that inhibits the enzyme that breaks it down. So I don't want to break this down. I could use that as a venom so like a poison, a toxin, or sometimes something that's a venom for one person is actually medicine for another. There are conditions where people's muscles twitch uncontrollably, and that's bad, and we want to stop the muscles from being stimulated by the nervous system. So something like a venom could actually be useful. What it's doing is competitively inhibiting the site where this molecule is binding. Now, the other type of an inhibitor is a non-competitive one. It's something that binds to an enzyme, but not on its active site. The idea is that when it binds to the molecule, or to the enzyme, even though it's not actually on the active site, it causes a structural change. So because it binds, something happens to the molecule, and the active site is different now. Good examples of things that do that are heavy metals. That's why they can be so toxic and so dangerous. The electron transport chain is something that happens all over the place. We'll talk about it really specifically in photosynthesis. I'll sketch the example a little bit bigger so that we can see it better. This didn't transfer nicely onto the screen. The idea is that I have an enzyme. I'm going to draw a really simple one because my drawing skills are poor. And I have a spot on it that's not the active site, but there's a spot where something else could attach. So this is the active site, but my molecule isn't going to bind there. It's going to attach to some other spot on the enzyme. When that happens, it changes the shape of the active site. So that now a molecule that used to be able to bind to that active site can't anymore because it's not the right shape. So that's the idea of a non-competitive inhibitor. It binds to an enzyme not on the active site, but when it does, it causes the active site to change shape enough that the regular substrate can no longer attach. Would anybody like to ask any questions about inhibitors? So then I'm going to give us a list of a few more things that could alter enzyme activity. And we're going to start just with a list. I'm going to put the list up, and I'm just going to ask you to think about how you think those things might affect an enzyme. And while people are thinking about it, anybody who needs to use the washroom, etc., this would be the time. So I'm just going to So I wrote up here on the screen 3 more things besides inhibitors that could affect the function of an enzyme. Now the first thing I'll mention is concentration. And when I talk about concentration, there's actually 2 kinds of concentration that interest us. Let's think about the concentration of the enzyme and the concentration of the substrate. Now on a previous page, the first one about enzymes, I said an enzyme can be used over and over and over again. So do you think that the concentration I have of an enzyme will have a huge impact on the overall result of my reaction? Now the answer here would be little effect. Little to no effect, and the reason is enzymes are reused. I don't just use them once and then they're gone. The same enzyme can act over and over and over again. So depending how much there was, it's possible that adding more enzyme will have a small effect, but there will be a maximum reached very quickly. And so the idea is that the concentration of the enzyme has very little effect on the actual overall function and how fast the reaction will happen. Now, what about the concentration of the substrate? Well, the substrate does not get used over and over again. every time the substrate comes into contact with the enzyme, reaction is happening. It could be anabolism or catabolism, one of the two types of metabolism. But the substrate concentration will have a great effect. More substrate equals greater rate of reaction. So it would make a difference how much substrate I have. If I add more substrate, more reaction can happen more quickly. So concentration has an effect if it's the concentration of the substrate. If it's the concentration of the enzyme, there's not a huge effect. Now we mentioned temperature and pH before, because temperature and pH are two things that can cause denaturation or coagulation. And so what I'd like to indicate is that too much or too little will have this effect, but there's always an optimum value. And that's the key point that I want to make. For example, if I was talking about an enzyme in your body, there is an optimum temperature. And very often we would represent this with a graph. So if I was talking about the function of an enzyme and I was talking about temperature and rate of reaction. Does anyone know what temperature your body is most of the time, or should be? 36 point something, it should be around 37. 37 is like what we would say is the standard normal thing. So if we were to graph the function of enzymes in your body, right around 37 degrees Celsius, you would see the graph go up. So enzymes in your body generally function best at somewhere around 37 degrees. They can still function if it's a bit colder or a bit warmer than 37 degrees, but not as well. And the reason is that as you change the temperature, now we're affecting the structure of the protein. Denaturation could possibly start happening. If I'm talking about pH, there are actually two examples that I'd like to give. You might be aware that in general, the pH of your body is somewhere near 8. It's not a perfect 7, it's a little bit higher than 7 in a lot of places. But there is a particular spot where the pH is more like 2. Some of the enzymes, especially in your digestive system, will have a graph that looks like that. They work best when the conditions are acid. That would be any enzyme that's in your stomach. Other enzymes work best when the pH is closer to neutral, when it's something like 8. Now we'll talk about specific enzymes later on, but the idea is that there is always an optimum value, where if you go higher or lower, it starts to function less well. But I can't give you a standard number because it's different for every enzyme. Now the last thing on the list is a cofactor or a coenzyme, and this is one of the things that we illustrated in the lab activity. A cofactor or a coenzyme is kind of like the opposite of an inhibitor. Can someone shut the drawer so I don't have to listen to that? A cofactor or a coenzyme increase the function of an enzyme. They do this two different ways, and it's dependent on where they bind. So both of them help an enzyme perform its function. The difference between them is where they attach on the enzyme. So a coenzyme is usually something that a vitamin does. It's not exclusively, but a vitamin is a really good example of something that is a coenzyme. A cofactor, a lot of the time, would be a mineral. A vitamin is usually an organic substance. It's a big molecule made with carbon. A mineral would be a smaller substance like sodium or potassium or copper. Coenzymes attach on the active site. Cofactors attach elsewhere. So very much like competitive and non-competitive inhibitors, the difference was where do they attach. Now you might ask yourself, well, you just told us if that happened, that was bad. How could this be good? And so I'm going to draw a little illustration. It's going to be fantastic. We all know how good of a drawer I am. Of the difference. Let's say I have an enzyme. My enzymes always look like Pac-Mans because it's the easiest shape to draw. Draw whatever you want. And my enzyme had a hole in it. And then my cofactor came along and filled up that spot. That's how a cofactor can help an enzyme. This looks a lot like a non-competitive inhibitor. The thing that's different is that in this case, the cofactor sort of completes the structure of the enzyme. It's not changing its structure, it's finishing it, making it better. A coenzyme would be something like this. Now let's say that this is my enzyme, and what I really want is a Pac-Man shaped active site. But that's not what shape it is right now. A coenzyme would be a molecule that comes in and fills in a space to make the active site complete. So it's still filling a spot in the enzyme, but this time it's making the active site the shape that it's supposed to be. So in this case, I didn't have a triangle-shaped active site. There was like a square hole underneath it. The coenzyme came in, filled in that space. Now my active site will actually work. Does anyone have anything they'd like to ask about that? So there's one more thing that I'd like to show you today because it's the topic of the lab that we're doing tomorrow. And it's how I could do a test in the lab to know what macromolecule I actually have. Do I have a carbohydrate? And there's a couple of different kinds of tests I can do there. Do I have a protein? Do I have a lipid? What kind of lipid do I have? And so the last thing that we're going to do is make a little chart that illustrates what test I could perform, what molecule it tests for, and what color change I could expect to see. Before I switch to that screen, is there anybody who has any questions about how I could affect the rate of enzyme function? So our last thing is tests for macromolecules. I made a little chart here. It'd probably be a good idea for you to make this same chart. Now on page 212 and 213 of your textbook, a lab is described where the goal is for you to discover this yourself. And I just want to make sure that everybody has the correct color results with the correct test, so that when on Wednesday we have a little quiz, which we will on Wednesday, you're sure which color things are supposed to turn. The first test that we'll use in the lab is called the Benedict test. It tests for, quite specifically, mono and sometimes disaccharides. So it's testing for small sugar or carbohydrate molecules. Now the Benedict's test starts off blue. That's its initial color. And then depending on the concentration of sugar that's in the substance you're testing, That blue will turn yellow, then green, then red, and finally brown if the concentration is really high. So that's one of the tests we'll perform in the lab tomorrow, the test for monosaccharides. The second test that's also for a carbohydrate is the iodine test. And the iodine test specifically tests for starch. Starch is a polysaccharide. And it's one that you would find typically in a plant. So potatoes are a good example of something that has starch in it. The iodine test starts off orangey yellowish, depending on how thick the solution is. And the final color is actually a really dark blue, but most of the time it just looks black. Now the third test is the Biuret, and I find that people mix up Benedict and Biuret because they start with the same letter. And so that's one that you have to pay particular attention to. The Biuret test is for proteins, and the Biuret test starts off blue and ends off a pinky purple color. Now, another way to say purple is violet, and if you say them out loud, Biuret, violet, they kind of sound the same. That's always been how I've remembered which one is which. So the Biuret test turns violet, which is how you could say purple, if there's a protein there. Now the Sudan-4 test is one that we may or may not do in the lab tomorrow, depending on what's available to me in the supply cupboard. But it is a test that's available. It tests for lipids, oils, fats. I put 3 different words there to indicate that anything really in that category it tests for. The Sudan-4 test starts off pink and turns red if a lipid is present. The one that we will definitely use in the lab is the translucence test. The translucence test is something that you've probably all seen in action before. You take a brown piece of paper, like a brown paper bag, and you put your substance on it. If it turns translucent, which means you couldn't see through it before and now you can kind of see through it, that's how you know you have a lipid, an oil, or a fat. If you've ever gone to A&W or McDonald's or Wendy's where they give you a brown paper bag, you have probably already seen a positive translucence test. These tests are the end of things that would appear on the quiz that we're going to do on Wednesday. So for the rest of this period, there are a couple of things that I just want to review with you, and then you can have some time to, let's say, look at the lab we already did, or read ahead and look at the lab we're going to do tomorrow.